आज ही डाउटनट ऐप डाउनलोड करें। डाउटनट पर होगा अब आपके सभी मैथ्स, केमिस्ट्री, फिजिक्स और बायोलॉजी डाउट्स का सफाया। बस अपने क्वेश्चन की फोटो खींचो, उसे क्रॉप करो और तुरंत वीडियो सॉल्यूशन पाओ। डाउनलोड नाउ।
There are two parts in this question. In part A, they're asking us to describe the various steps of Griffith's experiment that led to the conclusion of transforming principle. So, Frederick Griffith carried out this experiment where he worked with the bacteria that are responsible for causing the disease pneumonia. So, there were two strains of these Streptococcus pneumoniae bacteria. On one hand, we had the S strain. It was called so because it formed smooth and shiny colonies. And this was the virulent strain. Virulent means pathogenic. It was capable of causing pneumonia. Whereas on the other hand, in the same species, we had the R strain, which was called so because it formed rough colonies. And these bacteria were avirulent. They were not capable of causing pneumonia because they did not have the smooth polysaccharide coat that was found on the S strain bacteria. So what Griffith did during his experiments was, first he injected some mice with the S strain of this bacterium, and obviously these mice died of pneumonia because this was the virulent strain. Next what he did was, he injected some mice with the R strain, and since the R strain bacteria were not capable of causing pneumonia, these mice lived. Now in the next step of the experiment, again he took some S strain bacteria, but they had already been heated and killed. So by injecting these killed S strain bacteria, the mice did not die, they lived this time, because even though the bacteria was virulent, it was dead. But in the next step of the experiment, what Griffith did was, along with these heat-killed S strain bacteria, he took some live R strain bacteria also, and both of these he injected into the mice, and to his surprise what he found was that in this case the mice were dying. So what he concluded was that some or the other substance, some material had passed from this heat-killed S strain to the R strain, which made it capable of synthesizing the polysaccharide coat which is needed to cause pneumonia. So some transforming principle, some kind of genetic material must have passed from the dead S strain bacteria to the R strain, because from these dead mice, live S strain bacteria were isolated even though they had not been injected with any live S strain bacterium. So after this experiment, Griffith concluded that there was some kind of transforming principle, some kind of genetic material, some substance that, if passed from one organism to another, could transmit the hereditary information. So after this experiment, Griffith concluded that some transforming principle has been passed from S strain to the live R strain bacteria. But he was not sure about the chemical nature of this substance as yet. And this is what they're asking in the next part of the question. They're talking, how did the chemical nature of the transforming principle get established? Because most of the people were thinking that this transforming principle must have been some kind of protein. Some protein must have passed. So what they did was, there were three scientists, Avery, MacLeod, and McCarty, who carried out experiments to determine the chemical nature of the transforming principle, which was responsible for the transformation of the R strain bacteria in the final step of Griffith's experiment. So what they did here was, they used some enzymes. First of all, they took proteases, which were responsible for digestion of proteins, and then they also took RNases, which are responsible for the breakdown of RNA. So when digestion with these enzymes was carried out, in this stage of Griffith's experiment, they found that transformation was not hindered, transformation was still occurring, and the mice were still dying. So they concluded, even after digestion of proteins and RNA, if transformation was continuing, then obviously the transforming principle couldn't have been a protein or an RNA, because if it was digested, then transformation would not have occurred. So next they used DNase enzyme, and they found that after digesting with this enzyme, transformation was not occurring, and it was thus concluded that this transforming principle must have been DNA, deoxyribonucleic acid, which had led to the transformation of the R strain bacteria into the S strain bacteria, which made them virulent, and that is why the mice were dying. So the chemical nature of the transforming principle was established by Avery, MacLeod and McCarthy when they carried out digestion with certain enzymes. The digestion with protease and RNase did not affect the transformation, whereas after digestion with DNase, transformation was not occurring. So it was concluded it was DNA that must have passed from the S strain to the R strain bacteria during the process of transformation.
क्लास 6 से 12 से लेके नीट, IIT, जेईई मेंस और एडवांस के लेवल तक 10 मिलियन से ज़्यादा स्टूडेंट्स का भरोसा। आज ही डाउनलोड करें डाउटनट ऐप या व्हाट्सएप कीजिए अपने डाउट्स 8400 400 400 पर।